pleasure to introduce Dr. Julie Urban, who's an associate research professor at Penn State University. I had the pleasure of getting to know Julie Urban both through taking a genetics course that she taught when I was a graduate student there. Also, we crossed paths a few times as we were both working in extension and with invasive species, although they were different species. So Julie has a really cool background. She grew up in Toledo, Ohio, and went to undergrad at the University of Dayton. She then got her PhD in psychology from the University of Central Florida. And then she worked for a few years as a faculty member at a liberal arts school in Long Island. And then while there, she started becoming more and more interested in evolutionary biology. And so she actually decided to resign and get retrained. So then she earned her PhD in biology from the Ecology and Evolutionary Biology program at the University of Albany. And then before starting that position, she actually thought that she wanted to study the evolution of birds and song learning, but then she worked in a DNA lab at the New York State Museum for a summer. And that's where she discovered plant hoppers and became fascinated with them. So for her PhD research, she conducted work at the New York State Museum, where she collected plant hoppers from tropical regions around the world, and then used the DNA to reconstruct their evolutionary histories and their coevolution with multiple bacterial and fungal symbionts. So she's going to present a talk to you today on an update on spotted lanternfly research and its impacts in Eastern US. All right, Julie, take it off.
Thanks so much, Hillary, and thank you for giving me this opportunity to share some of my work with you today. I'm going to go ahead and share my screen with you and make sure you can see it. Okay, can you see that okay?
Mm-hmm.
All right, so this is the invasive plant hopper spotted lanternfly. And as Hillary mentioned, I started studying, I found myself in this area where now I'm doing very applied research. So since I've started working on lanternfly, my research I've continued my career trajectory and continuing to change a bit and learn and study new things. And so I've kind of made this bridge between basic and applied research that I think will be part of what you'll see in what I share with you. And so plant hoppers, why I became so fascinated with them, there we go, is that plant hoppers are, they're a really cool, diverse lineage of over 14,000 described species of insects. Here we have, just a sample of exemplars of different families. I have a couple of slides at the end where we'll talk about what families and what species diversity you have of plant hoppers in Maine, because Hillary shared a couple pictures with me and I was curious to see who you had. The plant hoppers, in terms of being crop pests, some of the major crop pests are in this family Delphasidae that you see here in the top left corner. These are little guys, 2 to 4 millimeters long, and basically they're the most diverse family of plant hopper. And they're major pests of all the cereal crops on the world. And so some of the most notorious pests are those of rice. We call them rice plant hoppers. But Niloparvata lugans, Sogatella crucifera, you might have heard of them. And when you get an outbreak of those in Asia, they'll knock down rice production by 80% or more. And so those are some of the more pestiferous species. And otherwise plant hoppers, there's a few pests and whatnot, but I kind of fell in love with them because of the remarkable morphological diversity you can see in color and body shape. And the relationship with symbionts, that we'll talk about in a little bit, they really fascinated me. And I never thought I loved an insect until I found these guys. And I said, okay, I'm finished. I'm going to study them. And so in doing my work as a graduate student at the New York State Museum, I got to travel around the world. My PhD advisor had a grant on Delphasids, so we traveled to the tropics, and I became interested in this one family in particular, Fulgoridae, or the lanternflies. And so this is where our spotted lanternfly belongs in terms of taxonomy in this family. And you can recognize this is actually a tropical plant hopper that I've collected a lot of in Malaysian Borneo. And you can recognize a plant hopper proper because they have their antenna beneath their eyes. They have like a carina or a frons in front of their eye on their face. And then Fulgoridae, many of them, if you're familiar with, perhaps you've heard of Fulgora or the peanut-headed bug, this Pyrops, a lot of Fulgorids are kind of trophy insects in terms of their unusual and pretty bright coloration, but also they have an extended head process. And this is really an empty structure. We don't know really what it does, but it was thought to harbor bioluminescent bacteria. That's the only hypothesis that's been tested about the head process of Fulgorides. And it was, no, it doesn't, right? But because it was thought to harbor bioluminescent bacteria and thought to glow, that's where they got the name lanternfly as a common name, but also even the name Fulgoridae as the name for this, as the nomotypic name for this family, but also for the superfamily of plant hoppers, named after the Roman goddess of lightning, Fulgora. So that's why they're called lanternflies. And so here, this is our spotted lanternfly. You can see, like any proper lanternfly, their beak or their mouth parts lay along their abdomen, and they use that to insert into the plant. And so you can see the mouth parts into a plant. All plant hoppers are thought to be phloem feeders. There's some nymphs we'll talk about in a little bit when we talk about the Maine plant hoppers that are thought to feed on fungus as nymphs. But spotted lanternfly is from Asia, and basically it was an invasive problem in South Korea in 2004. And there it was known to feed on a wide variety of hosts, as we see it doing in eastern US right now, but it caused the most severe damage to grapes. And so the primary damage, though, was it was from the plant losing its resources from the sap feeding of these insects, but also because they feed on a lot of sugary sap. They'll excrete a lot of sugary sap, and we call that honeydew. And then that honeydew is a substrate for sooty mold. And so you see that in aphids, you see that in other hemipterans. But in Korea, which was really interesting, that sooty mold that grew on the surface of the grape leaves blocked photosynthesis and was the primary basis of damage that lanternfly did in South Korea. And that's not at all what we're seeing here, which is an interesting story. So anyway, lanternflies are kind of weird among any kind of sap feeding insect that normally insects would lay their eggs on what the nymphs, who when they hatch out don't have wings and can't fly, and we think they can't move around. Usually you lay your eggs on something your nymphs can feed upon, but apparently plant hoppers didn't read that literature, or lanternfly didn't read that literature, and all lanternflies, or all Fulgoridae that we know of, actually do lay their eggs on essentially anything. So you can see here that these are lanternfly eggs that were laid on a rock. We think that they got here laid on a shipment of stone, either on the pallet or on the stone itself that was shipped most likely from China, but from Asia into a vineyard in into a stone yard in eastern Pennsylvania in prior to 2014. And so you can see lanternfly egg masses. Here they have this waxy covering on them. And here you can see some that don't have that waxy covering, or it's a little bit more worn away, where you can see it's really characteristic. The eggs are all lined up here in a row. Each egg case is contains between 30 and 70 eggs. And so one of the things, besides laying your eggs on anything, that's a good strategy for an invasive insect to move around a lot. But the other issue with lanternfly that facilitates its establishment as an invasive is not only that it feeds on over 70 different species of plants and trees, but one of its preferred hosts is tree of heaven, Ailanthus altissima. And so if you're familiar with the children's story, a tree grows in Brooklyn where a tree grows in a crack out of the sidewalk, that's tree of heaven. Tree of heaven grows everywhere. It was an introduced invasive here from the 1800s, I believe, but it essentially is itself just a mess. If you look at how many places tree of heaven has spread to throughout the country, it's ubiquitous. And in particular, in the case of lanternfly, we think how lanternfly is traveling and expanding its range is through hitchhikers, eggs, but also adults riding on rail cars, because rail lines are just filled with tree of heaven. And so rail lines and highways are big places where tree of heaven is spread and provide, this is one invasive providing an opportunity for another invasive to spread. And so the issue with tree of heaven, you might be familiar with it already, is that it grows through suckers. So if you chop it down, that doesn't do it. You need to kill it with an herbicide as well. And so this is a photo taken of a tree of heaven that has been cut down, and you see that doesn't stop lanternfly. They'll still keep feeding upon it. If you were to let this trunk continue without putting herbicide on it, it would shoot up little sprouts, and we call those candy canes. So because it's so hard to get rid of tree of heaven, it's kind of comparatively hard to get rid of lanternfly. And so basically, lanternfly was first detected in eastern Pennsylvania in Berks County on September 22nd, 2014. And at the time, I was not at Penn State. I was in my previous position at a museum in North Carolina Museum of Sciences. And because I had done genetic work, working, using DNA to reconstruct the evolutionary history of Fulguridae in particular, and I, was familiar with people who recognized what this was. They were also, interestingly enough, the guys at Pennsylvania Department of Ag who went out and did these initial surveys. They happened to have trained with one of the few plant hopper taxonomists in the country who was a close colleague of mine, and I knew these guys. And so, and I get a call from, this person who was a grad student when I was, and said, "Julie, this is confidential, call me back." I was like, I know Lycormas in the US, right? So, so basically they brought me onto the team early on to be part of the technical working group that USDA organized within those first 2 weeks, really, of confirming what this thing was. And myself and other people around the country, including scientists at USDA, APHIS, and ARS, started working on things. And so I started working on the genetics to try to see where it came from. And so what Pennsylvania Department of Agriculture did, and USDA, agreed to at that time, was to focus their treatment on tree of heaven. And so at this time, if we want to look at that history, back in 2014, the lanternfly was only found, when they did their surveys, in 6 townships. So these are the yellow regions here. You can see where we are. This blotched out area, that's this whole green area, is this blackened area in eastern Pennsylvania, just near Philadelphia, right? Oops. And so in that first year, lanternfly was only detected in those 6 townships, and they put out a quarantine in order to protect and restrict movement of goods and conveyances for lanternfly. From what they found in those surveys, they estimated, though, that lanternfly had been here at least 2 years prior to that, 2 to 3 years prior. And so what they did is they decided to pursue control through scraping egg masses, through putting up tree bands, which I don't know how familiar you are in hearing the lanternfly in the news and whatnot, but basically these are sticky bands that go around trees. And so when the nymphs climb up and down on the trees, it captures them in the glue. There's a lot of issue with these sticky bands, because if they're too wide, they can catch birds and vertebrates and all sorts of horrible things that people freak out about. So that's really bad. But basically, they did sticky banding to capture the nymphs. And then the main course of treatment was basically removing ailanthus, so like going into people's properties, getting permission, removing 90% roughly of tree of heaven on a property, and then using the remaining trees as trap trees. So what they would do, and this is continued treatment, is they inject those ailanthus with a neonicotinoid insecticide, dinotefuran, that acts as a systemic. And so the tree pulls that up into its tissues, and then any lanternfly who are kind of forced onto that tree in the area, is the logic, because you took everything else out, will feed on that dinotefuran and then die. So that's very effective. You'll see thousands of dead bodies, beneath these treated dinotefuran trees. But kind of the problem is that, there's a lot of lags. There's a lot of stuff we've written. I have a perspectives article and something else I'm doing now. By the time the government gets the contracts, gets the trees removed, get the trees treated, gets the money in line, and in this area of Eastern Pennsylvania, it's a pretty rugged area. And so it was pretty quick, within a couple of years, when it became clear that that management approach just wasn't keeping up with the population. And so the other issue here is, in the scientific community, does spotted lanternfly require tree of heaven to actually survive or to successfully complete its development? And the short answer is probably not. The longer answer is that's a really hard thing to study, which we can talk about more, because these insects are voracious. So nobody's been able to maintain an established colony of them. Tracy Lesky, who's a scientist at USDA ARS, has had the best luck so far, but it's a really hard thing to do. Why USDA and Pennsylvania Department of Ag focused and put all their eggs in that ailanthus basket is because if you're feeding on over 70 different species of trees and plants, maple, they don't like sugar maple, so I think you guys are kind of safe, but what they prefer is relative to what's in a landscape. And so when you have such a broadly feeding insect, if you went and shot everything up or sprayed everything with dinotefuran, now you're going to kill all your pollinators, you're going to kill a lot of non-targets. And so because it was thought that, ailanthus is an introduced invasive itself, so by focusing the control on ailanthus, that would be a way to reduce non-target impacts, which is why they focused on it. And so as you can see here in this graphic, the darn thing spread. By the end of 2015, you can see in orange, 9 additional townships were added, and they bopped into these other counties. And so that put the quarantine into 4 counties. By the end of 2016, they added 74. It was in a total of 74 municipalities, which are in blue, and that extended to 6 counties. And then in 2017, it expanded further. They got over this calling it by township or municipality and just called whole counties for the quarantine. And so it expanded in 2017 to 13 counties in Eastern Pennsylvania. But in 2016 is when I got the job at Penn State and got the opportunity to come here. So 2017 was my first full year. And so earlier in 2017, before this quarantine spread, we saw a few things that really set off alarm bells that showed that, like, oh, nuts, things are a lot worse than we thought. And so my graduate student, Erica Smyers, was doing some work on looking at insecticides and impacts on lanternfly in vineyards. And we were scouting out a vineyard right in that core infested area that she had been working at prior to that, for 2 years prior to that. And we saw, this is just one post of the vineyard just slathered with lanternfly egg cases. And this was throughout the vineyard. And so we thought, oh man, we've never seen this before. These look to be horrible numbers. And in fact, later in the season, we did see that. So here you can see, we had lanternflies coming in where literally you had thousands on one vine. And you can see I have a video that I'll show you later. I think I do in this, but basically you can see this wetness on the leaves. That's from the excreted honeydew. So we saw, especially in late season, we saw high numbers on grapes anyway, but then in the late season, they just really moved in and would solidly cover all the cordon and trunk of vines. So it was really alarming. But what was interesting is that in this particular vineyard where she had been working, it was a fruit orchard as well. So they were growing apple trees at a small road across from the vineyard, and the lanternfly had never ever been on apple trees. And one day, it was September 11th, 2017, and Erica called me and she said, Julie, I'm driving that road between the grapes and the apple trees, and they're hitting my car. We saw what seemed to be like this massive flight behavior that we really hadn't seen before. And so here, if you can see the video, this was what that looked like. Hi, buddy, little guy there crawling onto the screen. Okay, and so so basically what happened then, we saw, here's another example of what we saw that year. I've seen that every year since. Okay, so people freaked out, right? And so the public was really freaked out because the lanternfly, when they were flying, they were going into the Walmart, they were doing all this stuff. But also, even though we've been working with USDA and Pennsylvania Department of Agriculture, this got much higher attention at USDA. The technical working group had an emergency meeting held by the USDA. There were also joint Senate and House meetings, legislative meetings in Pennsylvania, because there was reports of damage in vineyards. And so that's kind of when everything notched up a bit. And so here, these are other scenes here from, one of those pictures of just showing you solid lanternfly. And then also we see, with all of that sooty mold growing on the honeydew, just the understory of forested areas, usually edge, like not into the forest, but understory areas were just like caked with black. It looked like somebody set fire to the plants. And again, you can see a lot of lanternfly. And so 2017 then was a year that kicked off a lot of research. There had been research going on prior to that, but it was all Farm Bill funded, which has been great. But that's funding that's provided 1 year at a time to scientists, and it's really hard to train your early career professionals, your graduate students, and your postdocs if you only have 1 year of funding at a time. And so I started pursuing other funding that it took 2 years to come through. But in the meantime, Penn State used money that is given to it in a line item kind of way from the legislature. They focused it all on lanternfly so we could get immediate research done in 2018 to provide, insecticide recommendations, to get, EE label amendments, and to do kind of those triage studies that were needed to be done, especially in terms of what products would be available for grape growers and for orchards and whatnot. But I'm gonna kind of, with that, if I don't want to really talk about all the insecticide recommendations, whatnot, all of that is kind of up on the Penn State Extension Lanternfly webpage. But if we look at where we are now, New York State IPM has a map that they update pretty regularly. And these blue areas are counties that are under quarantine. So you see that now Pennsylvania has 26 counties, including these 2 non-contiguous counties. This thing went through rail to get to Pittsburgh. It's also in Altoona. And so down here we see Maryland being impacted. What the guys at that I work with at Maryland Department of Ag, they tell me in this corner county, lanternfly is in every vineyard. And so you see also 8 counties in New Jersey under quarantine. Connecticut, there's an established population in Staten Island. And so pretty much what we're seeing here is that right now lanternfly it's in urban Philadelphia region. And so it's either in or moving into the major urban centers of the Eastern Seaboard. So I think, we see this thing spreading already, but I think it's going to be even further bad news. All these little purple points are where it's popped up. So you can see in North Carolina, Virginia, there's a population in Ohio. You can see it's all over, different places. It's popped up in New York. They don't have here, it showed up in a couple nurseries in Massachusetts, and from there got to New Hampshire. Again, they were all dead individuals and hitchhikers in this case. I don't think you've had any reports in Maine. Have you guys? Have you, Hillary, or you have? And so, yeah, it's a good hitchhiker. And so in terms of the damage and the impacts it's had, fortunately, like when we see it move into apple, and it's not just apple, it's like nectarine and stone fruit as well. It was reported, lanternfly was reported to be a pest of stone fruit and apples in South Korea. But when you really dig into that literature, it wasn't quantified. They only, like in the case of apple, the fact that sooty mold grew was how they was the basis for which they declared it was damaging. And so we're doing some studies to see if you put lanternfly on, and especially young tree fruit, does it cause the fruit to drop sooner or whatnot? But basically, we've never so far. They'll move into apples and stone fruit late in the season, feed for about 2 or 3 weeks, and move on. And there's been no reports of damage there. But unfortunately, grapes, that's not the case. And so here you can see this video. If you look over in this area, this little girl right here is going to be shooting out some copious honeydew, and you'll see what we're talking about. You can see some being shot over here in the top left. And so these things are just feeding voraciously. And they're really taking a lot from these plants. And we have a viticulturist who's leading some studies looking at what exactly are the physiological impacts on grapes. And we're working that up in a study right now. But basically it's even interesting. They'll move from the stems onto the cordons and down the trunks, kind of following the phenology of the grapevines as the grapevines are putting their phloem resources into the ground for storage. They track it, right? And so there's been another economic study that was done more recently, but back at the time in 2018, a survey done of looking at particular vineyards in that area. We see the vineyard where my student was doing her work, 90% yield loss that they declared in 2017, total death of another planting in another vineyard, yield reduction and whatnot. And in terms of where you really see where you saw the main economic impact in grape to start was the number of insecticide applications over the course of 3 years increased from 4.2 to 14, nearly tripling the cost of the insecticide. And that's still, not handling the problem. Okay. And so that particular vineyard where my student had done that work, by 2019, all 40 acres of grapes were dead. And so that's, pretty alarming. And the thing is, the insecticides will knock them down. And so if you see a spray of lanternfly in a vineyard, it's like a carpet of lanternfly beneath every vine. That's not the problem. The problem is more keep coming in. And so this is a vineyard where I did some work last year. And so here you can see, just damage to the vine. Out in this distance, what I wanted to show you this picture is that you see this area that looks almost like it's a lake, it's reflective. That's the roof of the Amazon distribution center, right, in Allentown, and a series of other like grocery store and other distribution centers. And so we have reports of lanternflies showing up dead. Fortunately, one was live this year, but all other reports were dead. But lanternflies showing up in these cargo planes in California. So you're talking, it's really a bad place for lanternfly to be in terms of this opportunity for spread and where it's getting to. The other issue, we see it actually damaging grape, but it doesn't, what it won't feed upon again, I said it doesn't really like sugar maple, but we've never really forced it. But what it does not feed upon are conifers. But that doesn't help so much because they'll lay their eggs on them. And so here we have a nursery that nearly went out of business because as lanternfly fly, you get, you get the bugs in your plants. And so those will need to be treated because in order to comply with the quarantine standards, they have to ship product that is free of lanternfly. And so, Penn State's kind of worked with them to make management recommendations, treating, doing activities during the night or early in the morning before they're flying, that kind of thing. But nurseries have been really the other one of the other main industries that's been impacted in addition to grape growers. And so here you can see when you'll just get waves of lanternfly coming out of these trees, and they'll just like solidly, hit those plants in this nursery. But then also Christmas tree growers, and you can see a couple of egg masses laid up in Christmas trees. And so what'll happen, you can see another one down over here. And so we've had reports from Christmas tree grower shipments. We had an external quarantine with New York where some Christmas trees were returned because they were inspected and found to have spotted lanternfly eggs on them. Now New York has their own infestation. Christmas tree growers have kind of, again, like nurseries, figured out ways to deal with this a little bit more. But now California has actually put an external quarantine up. So there's all sorts of trade implications here in terms of movement of goods. Anything that even a truck that could have an egg case has to be compliant with permitting that is in place. And then the other main issue here is that it's a huge nuisance pest to homeowners. And so you can see that, by some kids' toys here, this is a carpet of lanternfly because they fell down the chimney. And the other issues there is that, if they excrete honeydew on your deck, sooty mold will grow. So you can see this is a step that was power washed to remove the sooty mold that had been there. And so people are reporting, having a hard time selling their houses and whatnot. And then the other thing is that with the honeydew excretion and then some weeping that we see, especially in tree of heaven, not as much in other trees, but somewhat, is that you get this this is fermentation. These are fermenting yeasts and bacteria. I've sequenced them and they're full of yeasts and interesting stuff. But basically this stinky, smelly stuff and the honeydew attracts Hymenoptera. So people report not being able to go out in their yard because they're getting stung by these insects that are attracted to the honeydew. Okay. And so if we want to jump into the biology here, lanternfly, it's like, okay, why is this thing so hard to deal with? It's a fulgorid, for goodness sake. You have, we have, if we look at its life stages, it only goes through one generation a year. And so it lays its eggs, it overwinters as an egg mass in eastern Pennsylvania. In early May, it'll hatch out into these cute little first instar nymphs. They go through a series of molts where they have the first through third instars have the same appearance. They're just a little bit bigger. Fourth instars are red. Here they start to move on to woody tissues to feed. The little guys tend to feed on herbaceous tissues. And then we see adults emerge in eastern Pennsylvania by the end of July. And they'll feed and lay eggs until it takes a couple of freezes to kill them. And so, okay, why is this such a problem? Well, one of the issues is that if you're in your egg stage for basically 2/3 of the year for 8 months, and eggs can travel, that's a good long time for eggs to potentially move. On rail cars, we had a call this morning where there was a report in Michigan, and I think it was because people camped, people from the Philadelphia area camped in a campground in Michigan, and it was on their camper. Well, that's one of the issues. Adults last for a really long time. This plant hopper is the only one that any of us know about who study plant hoppers. And I've talked to this from colleagues in Europe and everything, that only has 4 instars and not 5. So it looks like it has that, an extended adult time period where it's a big bug and it's feeding a lot. And then the other issue that makes it really problematic is that the feeding preference of those life stages is relative to the host and their proximity. Like I was saying, we don't see it feeding on sugar maple because where it occurs with maple, it prefers red or silver. But if all you have is silver maple in an area, it might do, right? And so that even the other issue with this is that the nymphs feed on herbaceous tissues. The adults will too somewhat, but generally the adults feed on woody tissues. And so the issue here is that, yeah, a lot of sap feeding pests, feed on a broad range of hosts, but because these guys feed on woody and herbaceous tissues, they're moving across different habitats. So they're hitting agriculture, they're hitting urban landscapes, they're hitting suburban landscapes, and they're hitting, managed and natural forested areas. So moving across those different landscapes really makes it kind of a whack-a-mole problem to try to control. Okay, so if you look at, all of our control recommendations, all of the control recommendations are focused on the life stage. They say try to treat with ovicides or scrape during the egg stage. The early instars, put sticky bands up or knock them down. But here they're feeding on such a range of things, it's hard to really know where they are because they tend to be diffuse. Fourth instars will move on to woody tissues. They like to feed on black walnut, and that's kind of hard to treat because that's a nut producing tree. If Ailanthus is around in early season, they move on to that. And again, as I said, that's the basis of the governmental treatment efforts. But this late season movement, they'll move on to other things, and they're really hit red maple and silver maple hard. They move into vineyards then too hard. And so this is something that if we could predict better where almost like these reservoirs of lanternfly are going to move into, that might be a basis for control. And I've kind of gotten obsessed with this late season movement. And so again, I just want to point out that's a long time to live as an adult. They're feeding for those 4 months. And basically, Ailanthus, we don't know if some of this late season flight has to do with their mating behavior, but we know that when they feed on Ailanthus starts to senesce, and or the high numbers of lanternfly feeding on Ailanthus tap out those trees' resources, and then they move to feed. And so this is what I've been really working on here. And so here we see, if you know your lanternfly anatomy, you see the red valvulae here at the apex. These are both females, and I rip their legs off so you can see what's going on with their abdomen. But basically you see on the left is an early season female, and basically this female on the right was collected only 1 month later. And you can see when I say they feed voraciously, they get big and need to feed a lot for their eggs to develop. And so last year I just took live masses of males and females in a vineyard over the course of a few weeks. And basically in 4 weeks, females will increase their mass by over 50%. And so my interest here, back as the basic evolutionary biologist, one of the things I thought was so cool about plant hoppers was because they've co-evolved with bacteria. And so like other sap feeding insects, they're feeding on a sap-free or a nutrient-poor diet in their plant sap diet. And so they've co-evolved relationships with bacteria that provision them with essentially amino acids that's missing in the plant sap so they can make their proteins. And so other hoppers do what plant hoppers do, in that they actually organize those bacteria into actual organs or bacteriomes. And that's in itself a neat thing. So here you can see on the left, you have these orange strings are one species of the 3 bacteria lanternflies have co-evolved with. You kind of can't see it here, but there's like a whitish structure that's a second symbiont. And then here in this sausage-shaped organ is a third species of bacterium that I'm describing that's unique just to the lanternfly family. So these are relationships that have co-evolved these plant hoppers, over the course of tens of millions of years. And plant hoppers are the only known animals on the planet to make a different organ for every different species of bacteria that they've co-evolved with. And so this is kind of, I started tearing up lanternfly. I'm interested in this real, what I think is esoteric basic research, right? And so as I start dissecting lanternfly, if you look at an early season versus a late season female, I don't know if my little images here are in the way, but you can see that the shape, the position, and even the color of those bacteriomes changes as the females develop. And so it's like, okay, that makes sense. And I'll show you a couple more pictures in a second. That makes sense because as they get so big and their eggs develop from hardly anything to fully mature, those bacteria have to move into those eggs to get transmitted to the offspring. So, hey, maybe here, if we could disrupt what the endosymbionts are doing or disrupt that transmission, now that could be a really species specific way to hit lanternfly. And so that's what I'm working on. I'm doing some antibiotic and some chemical treatment along those lines. But a lot of what I'm doing is also just doing these dissections and looking at that progression of female development over time, because nobody's done that. There's only been one other paper on this particular species reproductive development, and that was from 1934. So you can see in the top left an early season female, her eggs aren't really developed. This is a picture I took of the spermatophore. It's kind of pretty. And so we can see a spermatophore here within the versicopulatrix. And here on the bottom right, you can see developed eggs. But basically, when we look at where the endosymbionts are in this early season female with the tiny little eggs, ovaries that aren't very well developed, again, this is all verified with DNA sequencing that I've done. You see these stringy structures here are the endosymbionts. But in a late season female, here you can see she has eggs that are nearly mature. The endosymbionts are these string things. So if you were to pull eggs out of this lanternfly, you'd be picking these symbionts out. And this is the leftover crystal of a spermatophore here. And so I'm working on, trying to figure out the timing of this reproductive development and the sequence of it to figure out how to mess with the transmission. But one of the things I've been learning in working with a big team of folks at Penn State doing this stuff is that, hey, what I think is basic biology really can have practical applications. And so I'm working with Dr. Dennis Calvin to develop a degree day model of female reproductive development. And so this is the basis of what we have right here. And this is that adult emergence period. And you see there's a pretty big time lag between when the females start to fatten up and when they actually lay the eggs. And so with this degree day model, we hope this is going to improve our prediction of spread and recommendations for that late season control. But it also might be, when we work out the degree day requirements, it might be that there's a northern limit, not in terms of egg survival, but in terms of northern limit to enough degree days for females to actually develop. So we'll let you know as soon as we know that. Also, other ways that basic biology is helping improve lanternfly control and management that I'm helping work on. We're using EPG technology to look at how lanternfly feeds and how they probe and damage different tissues. So this poor little guy right here is hooked up by a silver wire, and if you run a current through your lanternfly and have a probe in the plant, when the insect feeds, it completes a circuit, and so we can look at the waveforms. And what we see here is that first instars feed very differently than second instars. Second instars use what we think is a salivary sheath. And so basically, we'll be able to look at these different feeding waveforms and how they're penetrating plant tissue has implications for what and how plants defensively respond. So we're working with viticulturists to try to work at both ends against the middle to understand better exactly how lanternfly feeding is damaging grapes. Just blow through this really quickly. We started pursuing a grant where we could work at a regional level, that would be multi-year and whatnot. And so we just got funded for that in 2019. It's a $7.3 million grant, but and we have basically in-kind contribution from growers in terms of using their land to do this work. And so we have a lot of institutions in the region working on a lot of different aspects of biocontrol modeling, trap design, all sorts of things there. And so just mentioning, natural enemies, parasitoids, fungal pathogens, disruption of mating, lanternflies seem to prefer some trees over others. Also other stuff that Penn State is working on is, if can we use Bavaria or this fungal pathogen of insects to be like more of an organic pesticide approach to killing lanternfly and not have to rely so much on the neonicotinoid we've been, but also can we use broadcast applications, so treat a whole landscape and not just focus on one particular target trees. And again, looking at degree day models in grapes. So with that, I don't know if we want to go to questions, Hillary, or if we want to talk about plant hoppers of Maine. I'm game for whatever you want.
Talk about the plant hoppers of Maine, and we'll still have a few minutes for questions.
Okay, cool. Okay, so of the different groups of plant hoppers I used, I'll show you the book in a second. But Charles Bartlett's review of the plant hoppers of the United States. And Hillary shared with me one of the species you guys have is this cute little guy, Acanalonia bivittata. We have a place in upstate New York. And so like probably not as far from you. I'm just 2 miles from Vermont, an hour and a half north of Albany. And so I get these little guys all the time in my deck. Acanaloniides, that family. You have 53 species in 29 genera, and then one that Hillary showed me a picture of that wasn't in the list of the Bartlett book. And so you have Achiloidae. This is one of the species you have, Synodoscia timidata. Achiloids are one of the groups with durbids down here on the right, these little purple sedusa guys I get on my milkweed plants in New York. Both achiloids and durbids are thought to be obligate fungal feeders as nymphs, but they tend to feed on more herbaceous tissues. You have this cute little guy here, a calicellid, that you can see they're called pig bugs commonly. The genus is Bruchomorpha, and this is the species you have, Oculata. It's really short-winged. You have Sixiidae. These guys tend to insert their eggs into plant tissue. So they're 2 to 4 millimeters. You have a variety of genera and species there. It's no surprise that Delphasids are the most speciose group that you have. They're the most speciose group in general. These tend to be associated with grasses, but also sedges as well. How you can tell Delphasid is right here. You can see they have these little tibial spurs, like they're wearing little high heels. And so this is one, Sixiidae and Delphasidae are vectors, not always, right? And this is where they're a problem in rice and other crops. But basically, Delphasidae do their damage by vectoring viruses, and then Sixiidae vector phytoplasmas. Lanternfly is not known to transmit anything, but we're working on that to test it. Durbids, you can kind of see here, maybe if you get the presentation and zoom in or look, these are pictures all from Bug Guide. How you can tell a durbid is that the apical segment of the beak is really, really short. It's unusually short. Over here, how you can tell an achiloid is it's the only plant hopper family where the wings kind of overlap and they have that kind of flattened appearance. And then the sister family to Fulgoridae, so you don't have any Fulgoridae there. They say that Fulgoridae occur up into Maryland and Delaware in terms of historic records. If I want to get a Fulgorid, I'm going to have to go to North or South Carolina. They're harder to find. So you don't have any Fulgoridae up in Maine, but you have the sister family, Dictyoferidae. And so these little guys you might find on your goldenrod. And then this is one that wasn't listed. This is the Bartlett, O'Brien, and Wilson review of the plant hoppers of North America. It has a key to the families and the genera and distribution of all the plant hoppers in North America. So I would recommend that's your go-to for if you want to understand or learn more about plant hoppers of Maine. Charles Bartlett, he's at University of Delaware. He has an awesome website, literature and photos and things. So you can look up plant hoppers of North America there. But Hillary shared with me a picture that Jeff Normandin in your group had taken, and it's of the family Flattidae. Flattermanus proxima is my guess as to who it is. That's not listed in this book for Maine, but it is in the Northeast. And so it looks pretty much like that's who you have. And again, there's good plates in this book and online for Flattermanus. All right, and with that, I think that ends my talk.
Awesome, Julie. Thank you so much.
So did my share screen drop? So you have, okay.